O-Level Biology 5090, Paper 2, Variant 2 October November 2024, question number 1, 2, and 3. Question number 1 is use the dichotomous key in figure 1.1 to identify the 5 main vertebrate groups A, B, C, D, and E. So complete table 1.1, animals with backbones. Animals with backbones means these are the vertebrates. So now the vertebrates are further divided into two parts, that is the adults without fins and gills, and the other hand adults with fins and gills A. And you know the adults with fins and gills are only present in the fishes. These are the characteristics of the fishes, that they need gills and fishes because they live in water and they have to respire and they have to breathe during through water. So they need fins and gills. So these are the fishes. And we are moving towards the other side, that is the adults without fins and gills. Further, this group is divided into lay eggs with shells and do not lay eggs with shells. First of all, we will move towards the organisms which lay eggs with shells, have skin with feathers. Feathers are, only present in the birds. And have skin with no feathers. They have scales on their body, but no feathers are present. So they can be the reptiles because they also lay eggs. And the other organisms on the other side, which are do not lay eggs with shells, the organisms which do not lay eggs. So D, have skin with fur. These are the characteristics of the mammals that they have fur and hair on their body. So they are mammals and they do not lay eggs. With skin with no fur. Students, they do not lay eggs with shells. They lay eggs, but with no shells. Human beings, they do not lay eggs, whether they are with shell or without shell. Some of the mammals, they lay eggs, but you can see they have shells, skin with no fur. These are the organisms which do not have uh, proper egg shells. So they have membranous structure outside the egg. So these are the amphibians. So these are the five group of organisms. Now I'm moving towards the place where I have to write their names. So at A, it were fishes. And at B, they were birds with feathers. At C, they were reptiles. They don't have fur on their body, but they have tough skins in the form of scales. And D, they were mammals which do not lay eggs and they have fur or hair on their body. And at E, amphibians which lay eggs, but their eggs don't have the hard shells. So they are amphibians. So it was all about question 1. Moving towards question number 2. Here, a virus is a type of pathogen. Pathogen disease-causing agents are called as pathogens. So disease-causing agents are called as pathogens. Here you can see a virus and one labeling F. So this is the protein code. F is the protein code or protein envelope, and identify component F, protein code. You know, the viruses are the pathogens which cause different type of diseases, and these viruses are made up of protein, code, and DNA, genetic material, or RNA. If they have double strand means DNA, if they have single strand means RNA. So identify component F. Component F, this is the protein code, and these are the, the receptors. Uh, Next, identify one component of a virus not shown in figure 2.1. That is definitely the outer coat has been shown, but the inner core has not shown, and the inner core contains DNA or RNA, which has been not shown in this. As for example, if we see its internal structure, so you can see just like this, different. If they have single strand, so it can be the RNA. If they have double strand, it means they have DNA. So they have not shown this DNA because they have shown only the outer structure of the virus. So outer structure is showing only protein code. Next part of this question is pathogens may be transmitted by to humans either by or direct contact or indirect contact. So list two of the human body's barriers to the entry of the pathogens. And students, the pathogens have many ways to enter into the body. As for example, if they can enter through our nose, they can enter through our mouth, so they can enter uh, through a cut which can be on our body, or so in this way they have many ways to enter into the body, and these pathogens, when they enter into the body, they cause different type of diseases. So there are some natural barriers in the body which try to stop them, which try to make their entry not possible. That is, they sometimes try to stop them. So in this way, these natural barriers can be your skin, your body skin, uh, and secondly, your you can see the mucus glands which are present in your respiratory tract. So these mucus glands secretes the mucus. So these mucus glands in your respiratory tracts, they secretes the mucus. Mucus can traps the microbes, and same with these mucus glands, there are here in your respiratory tract. So we can also write here. So they captures the dust particles, plus they capture the microbes, and in this way they stop to them to enter into our body, by capturing them into the respiratory tract. And there is one more thing in the body, that is stomach produces have gastric glands, and these gastric glands secretes the gastric juice. And the gastric juice has HCl, and pepsin and enzyme. So this HCl is highly acidic. Its pH is 2. It has pH 2. And due to this, highly acidic nature, the proteins are digested into the stomach. Digestion of proteins. And secondly, this highly acidic nature helps to kill the microbes. If they are entered into our body by mouth with food, so in this way they are our natural body immune system or the natural body barriers help to kill them in this way. If a virus is, next question is, if a virus is able to pass through the body's barriers and immune response takes place, antibodies are produced during the immune response. Name 4 chemical elements that will always be present in an antibody molecule. Antibodies are protein in nature. They are protein in nature. Already, you can say, that these antibodies are protein in nature. So, the proteins are made up of different elements, as the elements which are common in all types of nutrients. They are carbon, second one is hydrogen, and third one is oxygen, and fourth one can be sulfur, nitrogen, but we will write over here nitrogen. So these are the major 4 elements which are present in proteins. Beside these, there can be the sulfur, there can be the phosphorus, or in this way, these sulfur and phosphorus, maybe they are present, but these 4 elements are definitely the part of protein molecule. Now, next question is, explain how the structure and function of any antibody molecule are important in an immune response. Any organism, any microorganism which enters into the body, when it enters into the body, it produces the toxins. So it produces toxins, and the toxins are very harmful for our body. So when they enter into the body, so our immune system start producing the antibodies. So these antibodies are produced as a result of the toxins or the element or the chemicals which are produced by the microbes. So antibodies have a V-shaped structure. In this way, I want to draw the V-shaped structure of antibodies in this way. So these are the V-shaped structure in which you can see there are 2 light chain, and these are heavy chains, these. And this is the region, this is and on both sides, the region is called as the antigen binding site. So these 2 chains, 4 chains are joined with one another with the disulfide bonds. So I'm going to write over here, that is how the structure and function of the antibody molecule are important. I have explained the structure. So I'm also going to write all this For your convenience, antibodies have a Y-shaped structure and already told you made up of four chains, two heavy and two light chains. I'm not going to write all this. So the tip of the Y-shaped form the variable region where the microorganisms will be attached. And you know, students, these are the antibodies has complementary shapes. And different types of antigens will be killed by different types of antibodies. So after attachment, with antibodies, with antigens, antigen antibody complexes are formed. Antibody, antigen, are formed. The constant region of the antibody can bind to receptors or phagocytes. And you know, the phagocytes do the process of phagocytosis for phagocytosis. And phagocytosis is the engulfing of the microbes. That is, the phagocytes engulf the microorganisms which are neutralized or killed by the lymphocytes. So in this way, the structure of antibodies directly relates to their ability to their to their ability to identify and eliminate the pathogens. Now we are moving towards the next question. A human mother may give birth to her baby before the end of the usual length of pregnancy. This is known as pregnancy birth. So, premature birth suggests why babies born prematurely are at high risk of developing some infectious diseases than babies born after 40 weeks of pregnancy. Students, when uh, there are 40 weeks of a complete healthy pregnancy. If sometimes the babies are born prematurely, that is, they are unable to complete their 40 weeks uh, as a fetus in the body of the mother. So they are at high risk of developing some infectious diseases because when they are inside the body of the mother, their mother's immune system save the fetus save the fetus from many different types of infections because they are at that time they are using the antibodies which are present in the blood of the mother. So in this way, they are safe when they are before birth. But when they have complete pregnancy time and at the after completed their pregnancy times of 40 weeks, they are uh, you can say born, and then at that time uh, they have no risk of infections. So these infections can even kill them. So these or even they can have the different type of diseases in their body. So the reason is that is they after pregnancy premature birth, they have very few number of antibodies because already they were using the antibodies from their mother. So lots of antibodies are present in the body of the mother, in the blood of the mother. So those antibodies save them from different infections. But when they are prematurely birth, they are you can say some they are you can say under different conditions. They are kept in this way to save them from different types of infections. So because they have few number of antibodies and they can there are reduced transfer of maternal antibodies, and so we can say there are few antibodies at that time set during the later stages of pregnancy. The mother transfers significant amount of protective antibodies across the placenta to the fetus. So I would like to say, but when they are, you can say, prematurely born, so at that time, the maternal antibodies are unable to provide them passive immunity to protect them against infectious diseases, so they might encounter in the first few months of life. So premature babies receive fewer of these crucial antibodies due to their early birth, leaving them more vulnerable. So transfer of reduced antibodies across the placenta. And uh, there is one more thing which we can see, that there is the lack of complete development of immune organs. One more, the lack of complete development of immune organ. So we can say the organs crucial for immune function, such as the thymus and the spleen, and may not be fully developed in premature infants, further compromising their ability to mount an effective immune response. So in this way, they are at high risk. Suggest one action that a mother can take to protect her premature baby from infection in the week after birth. So I would like to say that the mother can uh, protect her premature baby from infection in the weeks after birth. So she can, uh, you can say, uh, uh, protect them first of all by breastfeeding. And in this way, she is able to provide them antibodies. She is able to transfer the antibodies through breastfeeding through the mother's milk. Mother's milk contains antibodies which can protect the infants or the newly born uh, babies from infections. And secondly, the mother should keep herself very healthy. She can frequently wash their hands with soap and water for at least 20 seconds, especially before touching the baby, after diaper changes, after using the restroom, and before preparing feeds. So, in this way, we have completed the question number 2. We are moving towards the question number 3. And question number 3 is, use words from the list to complete the paragraph. They have given us few words with the help of which we are going to fill in these blanks. Glucose is transported in solution by the component of blood called, the liquid portion of the blood is the plasma. So the movement of the blood takes place with the help of plasma, because plasma has cells inside. They are white blood cells, they are red blood cells, and they are the cells. Blood, the blood is a tissue. So glucose is, when it is transported in the form of solution, it means plasma is the liquid portion of the blood. So we will write over here plasma. If the concentration of glucose in the blood increases above normal, and this is the natural phenomena, when the concentration of glucose in the blood increases after normal, above the normal, the hormone insulin is secreted by the pancreas, because at that time pancreas is activated, and the part of the pancreas, islet of Langerhans, they start secreting the hormone which is insulin. So insulin will manage the extra glucose. It will change the extra glucose in the form of glycogen. So the hormone insulin is released into the blood by an endocrine glands. Endo. Endocrine glands are those glands which are called as ductless glands. Ductless glands, which pour their secretions directly into the blood. They secrete different hormones and pour their secretions directly into the blood. Glands called the pancreas. This hormone promotes the removal of glucose from the blood and its conversion into the chemical called glycogen. The extra glucose is stored in the form of glycogen, and or it is with the help of endocrine gland, which is pancreas acts as a endocrine gland, and then it will secrete the insulin, and insulin will change the glucose into glycogen. Next part is figure 3.1 is a photo micrograph of a type of blood cells. You can see the blood cell. How can we identify uh, this is red blood cell? This is red blood cell because you can see that this is the no nucleus, non-nucleated. It is thick at the edges and thin in the center. Circular, biconcave in shape. So these no nucleus, and you can see thick at the edges and thin in the center. So this is the red blood cell. Next part of the question is, if the concentration of the glucose in the blood increases above normal and the not controlled, this can result in changes to some cells of this body. They may lose their specialized shape, become less flexible, stick together with many other cells of the same type. Suggest and explain how these changes will affect the body. So, first of all, I will explain the question that they lose their specialized shape. So many blood cells, they have specific shapes that are crucial for their function. For example, the red blood cells already shown you in the previous question, that is they are They are biconcave shape. In this way, this circular biconcave in shape, and which maximize their, so the shape increases the surface area. This shape increases the surface area. No nucleus, non-nucleated, it also this increases the surface area for their maximum function. So they are able to able to squeeze through the blood vessels, easily pass through blood vessels. So this is the reason. And secondly, they are very flexible, easily can pass through the blood vessels, um, if they are, and become so. In this way, they will not pass through any blood vessels, stick together with many other cells of the same type. If they are stick with other, you can say, if they are clumping, if they are clumped, this is the one, clumped, they are, so in this way, I would like to say, if they are sticking together, then they reduce the oxygen transportation. If red blood cells lose their biconcave shape and become less flexible, their ability to efficiently carry oxygen to the body tissue is compromised. Less transportation of oxygen, and then reduced blood flow. So due to which there will be an increased risk of damage in the body. So these conditions can be perfumed, it can lead to the inflammation. So we can see different problems can occur in the body. So after this discussion, I would like to write the answer on the given space. You know that uncontrolled high blood glucose, uncontrolled high blood glucose level can alter the physical properties of blood. For example, red blood cell are unable to perform their essential functions of oxygen transport and free flow within the circulatory system. It will effect, energy level, organ functioning, organ functioning, and increase the risk of serious cardiovascular complications. So it was all about question number 1, 2, and 3. Please like, share, and subscribe my channel. Thanks for watching.